Okay, so remember in the FASTA format, you had to have a greater than sign, and that you could put as much information, and that was the information that you would read about you about the sequence. Below that was the sequence, and remember it couldn't go over 80 characters. So the FASTQ format is a similar format, and it tells you a little bit about it's a representation of the read. So here is an example of a FASTQ format. So the ID of the sequence, kind of like that goes with the greater than sign. You put an at sequence ID will tell you that okay, a new sequence is coming up, and there you can put as much information as you want. And this is your read. Okay, this is that fragment that you sequenced, and the plus sign is a gap, and this for each of this there is a symbol. For each of these residues there is a symbol, and the symbol is a representation of how well is the symbol is a representation of what is kind of how what is the confidence you have in that. Remember, sequencing processes are prone to errors, right? And so, for example, you might have a fluorescence, or the fluorescence doesn't work at that particular time, so you might have a just an empty space, or you might have something where the color is not very distinct or is very faint, and the software makes its best estimate that, well, at this point in time it was a thymine that was added. Okay, and so the confidence levels of that particular, for example, let's look at this one, A. So the confidence level of A being in this position correctly is denoted by these symbols. And these symbols, do range over a pretty long range. So let's look at what these sequences mean. So in terms of confidence, in the sense that what is the accuracy of that particular um, nucleotide being in that particular position, this is the order we are going into. So the one which is a percentage is the highest order, and as you go to smaller letters, the small letters, etc., the tilde, etc., that, so for example, you can have very little confidence in that particular nucleotide being in that particular position. So as you can see, it's a pretty wide. It's not like saying, well, in 1 out of 5, one being high confidence, 5 being low confidence. Okay, so this goes over a pretty big range. Now, I wanted to point out that there's two particular errors. Let's say, for example, you were writing a computer program and to parse this information. Well, notice something, for example, that there is the at sign. The at sign, denotes the beginning of a sequence. The at sign also denotes some sort of a confidence score. Okay, now between the sequence of the read and the confidence scores, you have a plus sign. And notice something, the plus sign gives you a pretty high confidence score. So sometimes, there is a potential for an error. Now, a smart programmer can basically identify this, make sure that it is different, and make sure that it is, you can parse it. You can be able to say, okay, I know the difference between when I'm going to create a sequence ID versus when I'm going to create a confidence score. Okay, there are potential for errors, so from those confidence scores, which is a representation of the quality of the data, we are now going to now we want to quantify some of that. You want to make sure that you can actually give numbers. You can actually translate those symbols into numbers. And remember, it's a pretty wide range, so there is possibility of error. So the Fred quality score is a measure of identifying the correct base in the right position. So it really is supposed to be an adenine there. Okay, how confident are we? And the Fred quality score will give you something like that. So look at this picture now. This is a picture of the results, and what you see here is, let's say this is part of a read, and you'll see that you see there are bars. So this is a read. uh, each residue each nucleotide is differently color coded, and so let's begin to understand this. So now you have these bars here. What do these bars indicate? An example of a base has been given a very high Phred score of 50, and we'll see what those scores mean, indicating there is a 99.999% probability that this has been correctly assigned. So the lengths of these bars, you see these little bars over the length, are an indication of how well what this quality is. Now, you notice something, that the quality is pretty high, and then the quality goes down, which means we're not really sure whether these are really properly assigned. An example of a base that has been given a Phred score of 10. Okay, a Phred score of 10. Now, this green line is a Phred score of 20. So if you are in kind of like just about here, then it's a score of 20. And if 50 is 99.999% probability, then we'll see what that 20 is. A little chart is going to show up here. A Phred's quality of a Phred score of 10 indicates there is a 90% probability that it has been correctly assigned. Now here you see an N, which means that there is no assignment. Okay, so it's kind of an unknown. An example of a base for which no Phred score could be calculated, since the sequencer could not determine which base was present, and therefore is designated as an N. So for example, if it doesn't fluoresce, if you look at the picture and it doesn't fluoresce, and you expect something to be there, but it doesn't fluoresce, and you see an empty space at that particular slice in the fluorescence of pictures that you're taking, then it'll be assigned as an N. Okay, so this is an N. That means it's a no score. Now, look at this picture. This picture also, remember, these things are color coded. So for example, T's are red, C's are blue, G's are black, and A's are green. And so what you'll see here is a relative probabilities. So for example, I say this is a G, so this is a peak for G, and it looks like it's a pretty strong probability. Then A and A is pretty strong. Now, remember, this graph is literally gives you the probability of A, C, T, and G. As you can see, if it's a high probability, you'll realize that this is green, and the blue, red, and blue, this is black, and the blue, red, green are virtually not there, right? Again, you see the strong probability, strong probability, strong probability, until we come here. Now, this is predicted as a T, and this one is an A, and this is a G. But see, there is an overriding blue graph there, which means either of this could be a C. Okay, so these could be C's, and that tells you this it's a small probability, which means there could be something else. Now, while these are pretty strong G's, the next four are pretty, black peak, black peak, black peak, black peak. But for some reason, the probability of it being a T is pretty high. It's not non-existent. See, it's not non-existent, and that kind of tells you that then. So there's different ways of looking at it. You say, okay, well, you know exactly what these are, and you can see the probability of it being anything else is pretty low. But somewhere in the middle, something happened during that experiment. So remember, these residues are, these nucleotides are being added one on top of the other. So let's say you added 40 of these. So somewhere between 10 and 25, something happened, and where the probability of the right one is pretty low. So you might say, well, yeah, this is green, but it looks like the T probability is quite high. And then as you go to the and so you see that the green probability of it being an A is pretty high, but then this is A. Okay, so it looks like somewhere in the middle of the experiment it became low. Okay, so once again, if it's at this green line, it's 20. If it's way above, it's pretty high. And we'll see, well, why does 50, a score of 50 means it's 99.999. Why does a score of Phred score of 10 indicates that it's just 90%? Let's see why. Okay, so the Phred score is calculated by this value, Q, negative log negative 10 times log to the base 10 of. So this is a score, the quality score of a value P. Okay, and so this is a Phred score, and so let's see what that means. So if a Phred score is 10, it means 1 in 10, 90%. Base call accuracy. Base call accuracy is the accuracy of predicting that is where that particular nucleotide is correct. So the question is, how does 10 translate to 1 in 10? And that's 90. How does 20 translate into 1 into 100? Well, it's very simple. So 1 in 10 is 10 to the negative 1. Okay, 1 in 10 is 10 to the negative 1. So that's 10 to the negative 1 is, if you remember logs, it's negative 1, 10 to the log to the base 10 of 10, which is 1. So negative 1 times 10 is 10. So 1 in 10 means 90%. Okay, that's why this is a score of 10. Now, if this is 1 in 100, 1 in 100 means 10 to the negative 2. So that's negative 2 times negative 10, which is 20. Okay, so that's why the higher the number, the higher Fred's score is 99.9999%. Okay, so 1 in a million, that's 10 to the negative 6, 10 to the negative 5, 10 to the negative 4. But when it's multiplied by negative log, okay, so if you don't know this, look at some of the properties of log to the base 10, and you'll get an idea. And this is very important. Okay, it's very important to understand this Fred score. So when you say what does 30 mean? Well, 30 means it's going to be 1 in 1000. 1 in 1000 is 10 to the negative 3, which is negative 3 times negative 10, which is 30.